back to At the Bench, the Neonatal Physician Scientist podcast of the Incubator. I'm Misty Good, and I'm a neonatologist scientist at UNC Chapel Hill, where I also serve as the division chief. And I have the pleasure of co-hosting today with Dr. Betsy Crouch. Betsy, would you like to introduce yourself?
Yes, I'm happy to introduce myself, but I think a lot of the listeners know who I am now. I'm a vascular biologist and a neuroscientist. I'm an assistant professor at UCSF, transitioning to associate this summer actually.
Congrats!
Thank you. Yeah, moving along, with a lot of blood, sweat, and tears. So yeah, excited about that. I'm also excited about our guest today, Dr. Pamela Good from Columbia University. So Dr. Good and I first met when I was doing my sub-I in the NICU at Columbia, and there were some really special moments in that rotation. The time I thought I was going to be a pediatric neurologist because I was a neuroscientist, and I thought that in order to study the brain that you had to be a neurologist. And I'm so grateful that the Columbia NICU was such a supportive and exciting place that I started to, like, put it together and realize that I could do that amazing clinical practice and still be a neuroscientist, and it really launched me to do what I get to do today. So thank you, Pamela, for being, like, one of my role models at the time, and now we get to be colleagues, and we're really excited to have you. Do you wanna provide a brief intro to,
Yeah.
fast forwarding, 12 years or so
Oh my
since then?
goodness. That's just the nicest introduction ever. Thank you so much. My name is Pamela Good. I am also a neonatologist here at Columbia University, where, as Betsy sort of alluded to or let you all know, I have sort of done my training all the way through here. And then I'm also a physician scientist, and I study neonatal kidneys, specifically neonatal kidney injury, as well as the long term health effects of being born preterm and extremely preterm. With the focus on sort of using mouse models to study that. And thank you both so much for inviting me to chat today.
Well, we're excited to talk to you for many reasons. I think your career trajectory is has been, quite eventful in really good ways, and also we'll sort of talk about something just off the bat, which is that you don't have a PhD, and so you found your way into investigative science after your MD degree. So I think that's also a really fruitful path for both Dr. Goods on the call.
Yes.
You should probably have the disclaimer that we're not related,
Yeah, that's
but I
probably
do think
true.
you're amazing. I wish you were family, so.
No. Um, yeah, but, I think it's really useful to talk about that career, path too, because many people find their way into investigative science after they've completed their MD degree and go on to really, productive, careers. So, maybe we'll get started and just say, can you tell us a little bit more about your science and starting all the way back on what first inspired you about the kidney?
Yeah, absolutely. And I can talk a little bit too about the sort of MD track into becoming a physician scientist. And so, when I started in medical school, I was undifferentiated, other than knowing I was very much interested in pediatrics and much less interested in taking care of an aging population. And so, within everything that I studied there, I sort of was focused a little bit more on pediatric diseases. But the one spot that really pulled me towards adult medicine was actually adult nephrology. And here at Columbia, there's an amazing department of adult nephrology, and they even had nephrocardiology rotation. I might be mixing up what they called it back then versus what they call it now, but it was really taking care of patients who were in the cardiac or regular intensive care unit from a kidney fluid electrolyte balance perspective. And sort of then fast forwarding to my beginning to focus on pediatrics and neonatology, that really fit in with what we see in neonates in terms of understanding fluid and electrolytes and how that balances with cardiac and other disease processes. And so when I started thinking about my fellowship and my fellowship research project, that was one of the things that came back to me was that I had really loved studying kidneys in medical school here at Columbia, and that that could be something that would be really fun to study going forward. I also felt like sort of back in it was probably what, 2015, 2016, we were just starting to get some of the really interesting studies about the effects of prematurity on kidney health, both in the short and long term, and it felt like such an awesome area to dive into. And so during my time looking for fellowships, I started looking for research projects and collaborators, and then really landed with some amazing mentors and some awesome sort of prospects here at Columbia, and so that's sort of how I launched into this area. And one of the other stories that I really like to tell about why I love the kidneys is that in medical school, when we did the sort of histology studies and got to look at them under the microscope, they are just so beautiful and so cool looking. And I remember sort of seeing some of those pictures on that old amphitheater that we used to sit in, and just thinking this would be a great thing to look at all day, every day. And I still look a lot under the microscope at kidneys, and I still think that they're absolutely stunning. And so that has been really fun, and another thing that really pulled me into this area.
Can I ask if you love the kidney so much and were inspired to study it earlier, then how did you decide neonatology versus nephrology?
Yeah.
Good question. I think I loved the transitional physiology that all of us neonatologists love. I loved being in deliveries. The clinical experience there was so fantastic. I loved OB as well, and so this is that one area within pediatrics where we collaborate so well with obstetricians. And I loved sort of the acute care inpatient nature. And so, one of the other things that I've loved about neonatology is that we take care of the whole baby. And so then, of course, in research, we're able to sort of focus in on one organ system. But the kidneys regulate so much. And so it does still feel like, in some ways, I'm a generalist.
Yeah. A lot of that resonates with me too, thinking about the brain. But I mean, I will say I'm a little, I don't know if we want to talk about this now or later, but like dissatisfied with the, I don't know, therapeutic toolbook we have for, um, I mean, to give one example, AKI, right? uh, Weight, fluid restrict. I don't know. Are you thinking yet about a different future for that, or because we'll get into some of your papers, but I
Yeah.
really appreciate that your papers are thinking about the long term consequences of prematurity on nephron development, nephrogenesis. But what about the acute picture as well?
Yeah, absolutely. My R01 that I just submitted, is much more focused on acute kidney injury.
Cool. There we go.
Yeah. So
Yeah.
feel like you're a plant with that question.
Nope, not at all.
I think, of
I
course.
review no grants for NIDDK, just to put that out there in case, the neuroscientist and vascular biologist somehow got confused.
No, no. No, of course, yes. I mean, we need in the area of acute kidney injury with neonates, we need better diagnostic tools. We need better
Uh-huh.
treatment strategies. But I think in order to get there, you first have to have a better understanding of the pathophysiology. And sort of my working hypothesis all along has been that premature kidneys are not going to respond the exact same way as a term infant's or as an adult's kidneys, to all of the insults that they're encountering in the neonatal period. And so that's why during my fellowship, we generated a mouse model that we think does a pretty good job of simulating preterm kidneys, in
Love
order
that.
to
I love your
start to
mouse
jump in
model.
there. I
Yeah.
know. I feel like you've heard about it a lot through the years. I'm glad you still like it.
But it didn't exist, right? And, like, that's the whole amazing thing about being a neonatal physician scientist is you can generate these models for these conditions that are incredibly important and and other scientists have limited insight into exactly how to generate the model. Sorry, Misty, I cut you off.
No, I was just gonna say, and, like, while we know about your model, please share with our listeners
Oh, yeah.
about your model. Like,
Yes.
1, how you had the forethought to be able to develop it, and then, reproducibility and all those things that we love to see in mouse models. Talk about
Yeah. So it was a collaborative effort, with some amazing mentorship here. So myself and my primary mentor, Feng Ming Lin, as well as Frank Constantini, who's a developmental biologist here, sat down together and sort of looked at what had been done and what could be done. And then we had someone who was in our shared lab space, sort of in the team science approach, who also had a mouse that she thought could help contribute, and that's Mina Rao over at Harvard, and sort of together, we thought about how to generate a mouse that had low nephron number that simulates the low nephron number that we see in humans who are born preterm. And so we were able to use a triple transgenic mouse and use both genetic and chemical approaches. And what we ultimately ended up doing was inhibiting sort of the end of the development of the kidney. So kidneys develop through branching morphogenesis. We were able to take advantage of some of the signaling pathways that are involved in that process. Um, Breed mice, have them know exactly when they were pregnant, and then at the end of their gestation, either through chemical methods or through genetic approaches, stop the branching of the developing kidney, and therefore have mice who have like a 40 to 60% nephron reduction, which we think is a good simulation of what we're seeing in our preterm infants, although all of that data is based on human autopsy studies, which are imperfect in and of themselves. Um, but really, it was sort of a huge collaborative approach with lots of help from incredibly smart people who sort of knew how one might go about doing this. And we've been using both, within that model, there's a chemical and a genetic approach. We've been using them both for years, really since, 2017, 2018. And, they're going strong.
And do you have a preference of which model you prefer? Or
Mhm.
how do you select which model?
Yeah. The chemical approach has effects outside of the kidney, whereas our genetic approach really is specific to the developing kidney. And so I prefer the genetic approach. I find that, you always worry about having effects that are extrarenal that you're not even looking for. And we, of course, sort of screened the known possible side effects or known effects from the inhibitor we were using, and found that it was pretty good. And it's great if you're going to use it with another genetic model. But I prefer our genetic model overall. And I think, the things that we're doing now are mostly focused on that specific one.
Yeah, so maybe just to, jump in there, I was so pleased when I was reviewing your materials for this podcast and remembering that you did your K looking specifically at the effects of gentamicin, um, on on the model of the preterm kidney. And, for those of our readers who haven't had a chance to read your papers, or do you wanna kind of provide a brief overview of your findings and what you're thinking of for next steps?
Yeah, so we are primarily interested in neonatal acute kidney injury, and as we mentioned earlier, we are also looking at some of the long-term effects of low nephron number at birth. But for neonates, we had to come up with a neonatal AKI model, and there are a few out there. We extrapolated from those and from some adult studies, and ultimately landed on using aminoglycoside toxicity, so using gentamicin to induce AKI. And we are able to do that in mice when they're about like 1 to 2 days of life. So really trying to target that newborn period when we're giving gentamicin to our patients. And what we found
That's
was
so
that
funny because
I
I
know.
figured that you were using it because that's a that's a common drug that we use in the
Oh,
NICU, and it's really important to understand, the pharmacology and the pharmacokinetics. And I still remember when I learned about the dosing of
yeah,
gentamicin,
yeah.
right? That you give
Based on
this, like.
GFR and
Yes, exactly.
yeah.
And so yeah, I didn't realize that you were looking for an AKI model and then chose
Oh,
gentamicin as
no,
opposed to,
yeah, it
like.
was an AKI model originally. Yeah, that was how we landed on it. Now I'm so
Uh-huh.
glad that it worked out in both sort of on both
Yeah,
sides,
exactly.
right?
Yeah.
I mean, of course, we wanna understand how the interventions that we are doing in the NICU are affecting sort of all of our, or the entirety of our patients, in their incubators, et cetera. but yeah, we specifically chose it as an AKI model. And so we were able to extrapolate from older rodent studies, to dose it and to have the pups survive the insult. And ultimately, what we found was that there was much more severe injury in mice with low nephron number that simulate our preterm humans, compared to we have litter mate controls, specifically using the genetic model. And so we were able to look, based on what we presume is pure related to nephron endowment at birth, the effect of this commonly used medication. And there's more inflammation. There is impaired proliferation after injury, and ultimately there's the accelerated development of a chronic kidney disease phenotype. And I think when I thought about using gentamicin clinically in fellowship and beyond, I really thought, okay, so maybe you might get an acute kidney injury from it, but that likely, you'll recover and there'll be no permanent damage from it. And so, I was really interested to find that in those with normal nephron number, indeed they recovered and their kidneys looked pretty much normal, in adolescent adult age, but those with low nephron number had really, impaired structure, by that time.
That's awesome. Could you share a little bit more? Because I know you're on a K, right? Maybe you could tell us a little bit more about your K, and then, I'm curious, like, the gentamicin model, we do, like, obviously in the NICU, we do all these rule out sepsis,
Yeah.
Amgen,
Yeah,
and I'm
Yeah.
yeah.
curious if, like, repeated exposures, impact nephron number, and if you've looked at that for, like, the culture negative sepsis that
Ye
we're not doing anymore, but, like, you know,
Yikes.
um,
Yikes.
I think people still are,
Yeah, yeah.
so, like, 7 days versus, the 48
4
hours.
days.
Mhm.
Yeah, so in that, for my K, I had proposed to just do 7 days of, we do the standard rodent AKI dose, but since then, we've done a fair amount of sort of dose titrations to determine, how soon do you see an effect, really, again, still focusing on that neonatal period, excuse me, and so, we start to see effects by 2 doses. So 2 days of gentamicin. And again, I do have to state that these doses are much, much higher than what we're using in humans. It's 100 mg per kg, compared to, yeah, a ton less that we see in humans. But these pups are otherwise healthy, right? They're not preterm. They don't have other ongoing hemodynamic instability or other medications they're getting or anything like that. And so, um, I think that while it is a much, much larger dose, there is some truth to, or sorry, maybe cut that piece out, some truth too. There is still clinical relevance in this model. And we've scaled back the doses. We're in the process of sort of cutting it back to see, at what point do we start to see injury. But certainly, by 2 days of administration, you start to see evidence of proximal tubular damage.
Wow, that's so interesting.
Um, and so to coming back to the clinical piece of it, that's 2 days is of rule-out sepsis at birth because you're born premature, right? And so I think, that certainly when we started to see, some of those findings that, that's worrisome. And then, so in terms of the effect on nephron number, so we haven't, when the mice are this small, and we're doing the AKI studies, we aren't also counting their nephron number. We're extrapolating from like the larger population data that we did early on, that, showed they're roughly 40 to 60% nephron reduced based on the model we used. So we're doing it less to look at sort of the nephrons generated, in that sort of postnatal period and more like as a pure AKI study, if that makes sense.
That's great. I see Betsy thinking.
Yeah, I'm, like, running to catch up because, it's really good stuff, and Pamela, I was telling, Misty before you got on the call today that the kidney's really understudied, I think. I mean, everything in neonatology is deserving, compared to the correlate of the adult organ systems and the amount of attention that they receive. But the kidney, I think, especially is challenging. And as like evidenced by the fact that you had to create a model to study it. So it's really fun and also just not something that, that that like we have a lot of experience talking about. So yeah, so my mind is running to catch up. But so why, if you have
Yeah.
fewer nephrons, I mean, just because you have fewer of them doesn't mean that they're necessarily weaker or more sensitive, but
Right.
it seems like they may be.
So regarding why, that's actually one of the big parts that I submitted for my K. And um, what is the mechanism? Why, in
Mhm.
the setting of low nephron endowment, is there more severe injury? And one of our hypotheses is that with low nephron number, you may have increased uptake of gentamicin per cell. And so what we did was we used a fluorescent labeled gentamicin, and we injected. Actually, the it ended up being the dose that we give to humans in pups, and we were able to then look under a microscope and see how much gen is being taken up. And by counting the number of cells, you can see how much gen is taken up per cell. And it's about 30%, 40% increased gen uptake in each cell in the setting of low nephron number. And so I do think that while clinically we look for things like what's the gen trough level,
Mhm.
um, you know, it's the same gent that's going and being seen by each cell in the kidney. And so we're not measuring how much gent is being taken up in each proximal tubular cell. And I think that then the increased exposure per cell is causing increased toxicity per cell. And then that's why we're seeing more severe AKI in these mice. That's why we're seeing, impaired repair processes and an accelerated development of a CKD phenotype.
So how would you measure that? Non-invasively?
In
Or
a human, I know it would be really
yeah.
hard to measure non-invasively, I think. Yeah, I don't know how, even how do we measure kidney function in preterm infants, right? You're measuring
Mhm.
serum creatinine, that's a terrible, way to measure it.
Yeah.
And so I think it's really hard. And so, I think a lot of people ask me when I give talks about my research, how has this affected my practice? And I think one thing that's really changed for me is to when I see a patient to actually try and think about like what do I think their nephron number might be compared to the average person, knowing that some of that is genetically programmed. Adult humans have a very wide range of nephron number, but some of it's related to things like were they growth restricted, how preterm were they,
Mhm.
and those who are extremely preterm, those who are growth restricted, I do think I think extra hard about using gentamicin. Um, and we just don't have the clinical data to be able to do it in an evidence-based way, because
Yeah.
again, this is all preclinical, and mice are not humans, but they're a great model.
I mean, they're the best model, in fact, because they're the only model that exists for, so for prematurity restricted, I don't know, nephron nephron diminutiveness.
Yeah, yeah, yeah.
How do you say it?
I mean, you can say low nephron number. Sometimes I say in, underdeveloped kidneys when I'm talking to patients and things like that, but yeah.
Yeah, I was just being
Is there
nerdy
anything that you
silly.
can do to, enhance nephron development in mice?
Yeah, it's a great question. I'm trying to think, there are studies showing that, things like, and I'm gonna blank on exactly which study it is, but things like prenatal steroids and things like that, encourage development, but we don't really have anything that will increase your nephron number, which is ultimately, I think, what many people who are interested in neonatal nephrology are concerned about in preterm infants. And then particularly in the extremely preterm population, right? So, over 50% of nephrons are formed in the third trimester. So, less than 28 weeks, you're looking at pretty significant deficits. And then when you push down to 22, 23 weeks, that's extreme nephron deficits would be expected. Um, and so, we don't have a great way to do that. Now, if we did, that would be amazing, and that would be something we would, want want to disseminate and share with the world, but not that I'm aware of now.
What about, is it known that our dosing of gentamicin is there any room to decrease our dose? in terms of its effectiveness with, killing gram negatives?
Yeah, so I think, a lot of the dosing, is based on the pharmacology, and then on expected changes in kidney function and GFR over time. So we had proposed, when my first case admission, we really wanted to look at gent levels in these mice, which I think would be outstanding. I've really struggled to do that and actually just got a lead from someone on trying to outsource it. But it would be done like using mass spec, for example.
Yeah, cool.
But there's all, we have to just make sure we have a machine that can measure gent levels and that can take mice samples. And then we also have to, as working with small mice, both of you, that it's always a volume issue as well. But that's
Yeah.
something that we definitely need to do, because that would be really helpful to then be able to translate back to humans.
Oh, yeah, when our mice with neck get sick, like, they have, like, after they've been euthanized, they have these big bladders, and so, if you ever want me to save any urine
Save
for
me some urine. Yeah, for sure.
kidneys or anything, just
Yeah.
to, evaluate, we used to, like, deflate the bladders because they were just in the way, you know?
Yeah, because you probably couldn't get at the rest of the intestinal contents, right?
Yes.
We can get urine from the pups, we do, but it's again, there's issues around, do you need to pool it? How much can you get per mouse? How many times can
Mm.
you sort of coax them to try and pee for you? Um, but yeah,
Yeah, and I think another thing that you do really well also is distinguish male and female effects. So, that also opens up just, so you've got control, you've got your genetic model, and then you've got male and female, so you've already got
yeah,
many groups to look at. So,
yeah,
yeah, complexity. So, yes, I think, for the listeners, we were talking a little bit about, so you're in your K now, and you just put in your R, which is excellent. Um, yeah, that's about the right timing.
yeah.
And then Misty was giving us a tip about other grant submissions that are available to you.
Yeah, I'm happy. I'm happy to talk about that for our listeners. So, something that I was really grateful for when I was on a K award through NIDDK through the NIH was this art free that is specific for K awardees. And so, for our listeners, prior to the show, I was just asking Pamela about if she had applied for that and or if that was on the horizon and what your thoughts were about that. But I think it's a amazing type of mechanism that it's 50,000 per year. So it certainly is not the equivalent of an R01, but, I do think it's really important for listeners who are on Ks, through particular institutes that do have that availability to be able to apply for those because that's, additional funding that can go for, obviously, reagents, and you don't need salary support when you're on a K, but it's you can put it towards a technician, or mouse costs, etcetera, so
Totally. Yeah, and sort of as we had talked about a little bit before, it's on the list. We made a big push for submitting an R this past cycle, but yeah, it will likely go in in the spring or summer. And there's always, the K, it was such an amazing opportunity to get the time to do these studies. But sometimes when I go back and look, I'm like, oh my, look at all these other things that I proposed that I was going to work on that I haven't quite gotten to yet. So,
Yeah.
I think it would be a great opportunity to be able to sort of look at the pieces that are still left and sort of expand on them and get some support for that. And every dollar helps.
especially in this funding climate, right? It
Yes.
does, and, it helps keep things going, and I remember when I was going through this, writing the first R1, I mean, props to you because it is hard, and, I think going for it is very, very admirable. I remember having a lot of pressure on me to, apply for the R1, apply for the R1,
Yeah, yeah.
and, I was very resistant, so, it all worked out, thanks, but, yeah, applaud you for going for it.
I mean,
Really.
yeah, it was definitely daunting, but, I think, it's helpful to have collaborators, mentors, and people who are willing to read with you and give you feedback. And then, just a shout out even to our grants administrators here who did such
Mhm.
an amazing job of sort of walking me through everything that needed to get done so that I could focus on the science. And I think that made a huge difference for me for this submission, just because it did feel incredibly overwhelming originally. But yeah, I'm sure the next time around when I revise it, it'll be less overwhelming.
I think
Yeah.
I think it's less, but also, yes, shout out to the grants administrators out
I was gonna
that's like
say, I think we should have a session dedicated to the grants administrators for this, segment of the At the Bench specifically,
Yes.
like
Oh my goodness. If I look at my sent email, I think there's probably like within a day, so many. And they were there, they've submitted so many grants. They really, our staff here knew what they were doing and
Uh-huh.
were so kind throughout the process. I really, really appreciated that.
Oh,
they're
man.
experts at that. I
Yeah.
always treat mine with, after we submit something, it's usually, down to the wire. I'm not one of those people that submits really early, and so I always get them something, whether it's like a Starbucks gift card to say thank you,
Yeah.
and so I prime them for the next, stressful event, which may be the next cycle, and
Oh, that's a great tip. Yeah.
I have this amazing story with my grants administrator who is, Beth Bartlemez, and I had a situation this past fall where there were some layoffs at UCSF in the pre-awards, division, and it included the staff who previously had been submitting for the grad students because now it's kind of like each department doesn't have its own people, they're all centralized. So they laid off, like, the person who was supposed to submit my grad students, fellowship awards, and then there was a supervisor who supposedly was available, but in fact, like, just didn't respond to any emails.
Oh
So
no.
it was a half an hour before my students had their like, because we had been emailing with this person who had an out of office message. Like the person who was supposed to be submitting, for the people who had been laid off had an out of office message for the day that the grant was due. And we were just thinking, how do we then submit the grant? So I called my person, Beth, half an hour before it was due and said, "Beth, I'm in a pickle. It's not your pickle, but I'm wondering if you could help me with
No.
my pickle."
Mm.
And she goes, "Yes, I was aware that this could be a problem. Just assign me as the institutional person and I'll make sure it gets done."
Oh,
And she
that's
that's
did.
great.
amazing.
And one of my students got it.
Wow. Congrats.
That's my most extraordinary story.
That's
Wow.
incredible, but also that deserves a reward.
Yes, I did. I yes. Yes, I also nominated her for, like, a UCSF-wide award for that particular, example of excellence, but
That's
there have been other
saving the day. Like,
yeah, I mean, literally, right? So, anyway, but it's also nice to honor her, so thanks for giving me that opportunity. Well, this has been a blast, and, we always like to end on something that's fun about our guests and their research teams. So, Pamela, do you wanna tell us about something that you do to relax or, to teamwork with your other, colleagues?
Absolutely. And actually, before we end, I realized, earlier when you mentioned, the kidney and being understudied in the field of neonatology, I do want to also give a shout out to the Neonatal Kidney Collaborative,
Cool,
and
yeah.
also to tune in, especially our trainees who are on listening today to this organization because it's a group of pediatric nephrologists and neonatologists who are working together to fix that exact problem. Both in the realm of education, patient advocacy, and research. And so that group is outstanding. And the trainees, there's awesome educational information on their website. So can direct you that way if this was inspiring and you want to also jump into the world of neonatal nephrology.
Or I think just to
Um,
brush up,
yeah,
and,
exactly. There's lots of wonderful resources, including some of the more recent excellent papers
mhm.
within the field. So, but to get back to your question, so our lab does do some fun lab lunches, and one of the things that I started doing this year was asking. very steadily what the favorite desserts are for the individuals in our lab. And then that has also sent me sort of, on an exploration of amazing dessert places in Manhattan, of which there's so many. And so I've tried some new cakes and some really fun new things that we've never had before, including like a miso carrot cake last time around, which has ended up coming from the bakery down the street from my apartment. So I think it's fun to have dedicated time with your team that's not talking about science at all. And so this year we've really been trying to do that. I'm sure you guys have already knew that trick and have been doing it for a long time, but we've really enjoyed it. And then I have, as Betsy knows, I have 2 small kids, and they have, they've also been able to sort of come and meet people in the lab, and I'm hoping to inspire some future female scientists. I have 2 girls, so
Oh, that's awesome. Speaking of inspiring, what would you like to share with our listeners who are maybe going through it, or what have you told yourself to keep things going when times get rough?
yeah, that's such an important question. I think one thing is what I found personally is when I got unexpected or disappointing results or failed experiments, it's pretty easy to sort of be focused on that one failure, and to get really down because we're really invested. We've put so much into these models and these projects. So one thing that I like to do in that scenario is to sort of like take a step back and try and look because there's lots of other things that we're all working on and doing. It's usually multiple projects. There's also the clinical side, the education side as a physician scientist as well. And so when I'm in one of those moments where, I've had, the 10th failed staining and I'm really bummed that I can't get an antibody to work, I like to take a step back and say, okay, but like, let me look at the past, 2 weeks, at, our lab meeting agendas and look at something that was successful there. Or even if there's nothing there, like, look back at the last time I was on call and think about, an opportunity that went well in the clinical world. I think that's one of the great things about being, a physician and a scientist is that you have the opportunity to be in all of those worlds. And when things aren't going well in one area, you can always sort of refer back to the other area, or even just think back to the patients that you're taking care of and remember why you're studying, neck or the brain or the kidney.
I
Oh,
love that. It's important to know why, right? And
yeah,
circle back when you're slogging through and things aren't working. you know?
totally. And remember that, that if you could do it in one experiment, this would be easy and everybody would do it and things would move super quickly. And so you also have to sort of dig back into that sort of resilience that we all worked on so much going through training and keep trying.
Absolutely. Well, that's terrific. I love that
Yeah.
advice.
Thank you, Pamela, for your time and for that really fantastic ending. To our listeners, thanks for joining us and look forward to catching you at the next At the Bench.